Good afternoon. It's my pleasure to introduce Dr. Nesvizhskii. Dr. Nesvizhskii serves both as Godfrey Doris Stobie Professor of Bioinformatics and as Director of Proteomics Resource Facility at the University of Michigan, Ann Arbor. He is founder and CEO of FragPipe, which leads efforts to commercialize computational algorithms for mass spectrometry data analysis. He also serves as senior editor in bioinformatics and biostatistics for the Journal of Proteomics, sits on the editorial board of Molecular and Cellular Proteomics, and is a member of the Scientific Advisory Board at the Swiss Institute of Bioinformatics. Please join me in welcoming Dr. Nesvizhskii.
Well, thank you, Nikolai. I think all the organizers for inviting me to come here. It's my second time at this conference. I have to put some disclosures required for me to put here related to conflict of interest and financial interest related to licensing of our tools, MS-Fragger, IronQuant, and DiaTracer through our company, Fragmatics, as well as other relationships here. So FragPipe is really a great computational platform because you can run it on the cloud, you can run it on a cluster, on a desktop. We have about 14 to 15,000 downloads for each version. We just released a new version, FragPipe 23. And it's free to academics, and as I already mentioned, it's commercially available to industrial partners. So before I get into the single-cell proteomics, I give a little bit of a brief introduction to the tools themselves. But why? One advantage of FragPipe is that we have this system of workflows where you could basically load the workflow of interest, whether it's a DIA with spectral library building, direct DIA spectral library building at quantification, or it's a TMT or SciLac, chemoproteomics, immunopeptidomics. Just load the workflow and everything is going to be set for you. You just need to load the workflow, the data, the database, and click run. And what I'll talk today about some of the workflows that involve single-cell proteomics data, although the same workflows that we use for single-cell, you would use for other data sets as well. So we don't have actually single-cell specific workflows because we found that most of the strategies are actually pretty much the same as for bulk proteomics data analysis, as you'll see from my presentation. We also have a FragPipe Analyst, a downstream tool that's super convenient for you to quickly visualize results of your quantitative workflow, again, whether it's a DIA or MS1 with match between runs or TMT workflow. You can quickly visualize volcano plots, PCA plots, do enrichment analysis for experiments that involve differential expression analysis. Perhaps not for single cell so much, where you use dedicated tools like Sam is developing here, but for a lot of standard differential expression analysis workflows, this is a very, very powerful tool. So some of the updates that are relevant to this presentation in FragPipe is that we have a new tools for DIA data analysis, a new DDA plus tool for chimeric peptide identification from MS spectrum. We have deep learning-based rescoring. We have visualization tools, like I said. In addition to FragPipe Analyst, we integrate with Skyline, MSStats, and we have a lot of new PTM workflows and glycoproteomics workflows and so on and so forth. So a lot of different things. But I will get now more specifically to things that are related to single-cell proteomics. I actually like really the single-cell proteomics community because it's really pushing the envelope and helps us computational scientists to develop new tools and new strategies. So even if at the end, the way how we analyze the data for single cell is not necessarily that much different for analyzing bulk proteomics data, it's actually pushes us to optimize the workflows because everything that we optimize for single cell benefits pretty much every user. So we need methods that work with single cell as well as with bulk proteomics data sets, ideally through the same workflow, the same pipeline. And we have to pay extra attention to things such as match between runs and false discovery rate analysis related to match between runs, especially when you use bulk data or 100-cell data to build the libraries boosting runs and then transfer the identifications to single cell runs, and some of the additional challenges with how we do deep learning-based rescoring, or some of the new TMT strategies that people explored for single-cell data. So things like this really pushed us to develop new things in a pipeline that, again, at the end benefit pretty much everyone using the tools, not only single-cell community. So, for example, one thing that we were really pushing hard is this development from MS Booster rescoring tool that utilizes deep learning-based predictions. So what's shown on this slide here is a typical workflow where you would identify peptides from MS spectra using a search engine. In our case, it's MS Fragger. And then it would go to percolator for rescoring, and then downstream protein inference analysis and so on. But if you can predict retention times and spectra using deep learning-based predictions and use those predictions in scoring, you can significantly boost your identification rates in challenging data sets, right? In the regular trips and digest data sets, you're not really going to gain anything. But for challenging data sets, and we thought that single cell would actually be one of those data sets, and indeed, this is where it helps, that if you include deep learning-based predictions and incorporate them in scoring, that would improve the number of identifications. And MS Booster is our tool that's doing exactly that. And again, single cell analysis was one of the motivations for developing these tools, actually. So MS Booster basically will predict, as I said, retention times, spectra, and ion mobility values if it's a timsTOF instrument, and then would add additional scores based on how concordant the predicted retention time and an empirical one, how well they agree. So a peptide with a big difference would be penalized, and the same for the spectral similarity. Initially, in the early days, we thought that actually we would have to retrain the deep learning model specifically of single-cell data, and Sam had a paper that I think alluded to that point, that single-cell spectra would be would look different from bulk proteomics data. So kind of now it's really depends on the data set. I would say that we see some single-cell proteomics data sets where conventional predictions work well, and we see some other data sets where spectra look very different than what you predict using existing predictors trained on bulk data. So in general, we do not, for example, on our tools now use any single cell specific sort of deep learning model for predicting spectral retention times. And still we get 5 to 10, sometimes 15% increase in the number of identifications by utilizing predictions when applied to single cell data. So here is, for example, one data set where if you look at the spectral similarity between the predicted and experimental spectrum for the single cell run versus 5 cell versus bulk cell, you see that the spectral similarity is pretty much the same, even for single cell data as for bulk data. But as I said, in other data sets, you'll see that the similarity between the single cell run and the predicted spectra would be much worse than for bulk data versus predicted spectra. So it really depends on the data set. So, but to make it more robust, because we don't want to be sort of at the mercy of how the spectra look like retention times in a particular data set, to benefit no matter what, we also work on creating a system where you could actually utilize multiple different predictors. And we worked with Matthias Wilhelm lab on this Koina server. Koina is a server that hosts multiple deep learning models for retention time spectral predictions from PROSIT, from AlphaPepDeep, from several other tools. And the idea of Koina is that in your pipeline, in our case it's FragPipe with MS Booster, you could send peptide identifications, you can send peptide sequences and spectra to this Koina prediction server, and basically try every model there. And we automated in FragPipe in MS Booster, so we send a little bit of data, not the whole data set, and test all models that's available on Koina. And then we find the one that for the particular data set we're analyzing gives the best similarity and the best retention time agreement. And then for the full analysis on all data, then we are using that best model. So we basically created this easy-to-use infrastructure for finding the best prediction model. So even if one predictor doesn't work well on a particular data set, probably another predictor from another tool would work better. So that improves the robustness. This being said, again, I don't think there is any predictor that was trained specifically on single-cell data, but I find that with this Coinage server now, especially in multiple models, we actually get robust results even for single-cell data sets, no matter what the data set is. So it improves the number of IDs for single-cell data, but it also improves in other applications, for example, HLA peptidomics, and we do ton of sort of collaborations in this HLA immunopeptidomic space, and their predictions are also very useful. So basically, any type of experiment where there is a large gray zone sort of between true and false identifications, a large source space or challenging data, this type of predictions have the most impact. And the regular trypsin digest, it's minimal. So quantification, and then we again worked a lot over the last 5 years on developing tools for quantifying peptides with the best accuracy and precision possible, whether it's a MS1 or a label-free MS1 or SILAC or TMT-based workflow or DIA. So we worked on it. We developed a tool called IonQuant for MS1-based quantification with match between runs. And actually adding match between runs and IonQuant was one of the motivations is single-cell proteomics data. Because when you build, and it's the same with DIA, and Vadim actually adopted a lot of, I collaborated a lot with Vadim Demichev, and he also adopted a lot of the concepts in match between runs for DIA there. So the concept is that you can analyze your single-cell data along with your boosting runs, library runs, right? Maybe 10 cells, 100 cells, if not even bulk data, which is not ideal for sure. So you have these boosting runs. So you identify a lot of peptides from those boosting runs because it's a lot more starting material. And then you build a library that has a lot of peptides. And then you go back to single-cell runs and you look for those peptides, right? Now, so whether you do it with MS1, with match between runs, or you do it in DIA, where match between runs is sort of, inherently built in the tools, it's the same challenge because you can FDR control the library well, but you go back to each run and you're transferring still a lot more IDs from your sort of bigger library to each individual run, and you have to control the error rates at the run-specific level. So for MS1, we developed this match between runs in IonQuant, and this is part of these papers here that we described. And I'm not going to talk too much about MS1 now at the moment, except I'll say that in FragPipe 23 that we released just a few weeks ago, we actually made a lot of improvements in IonQuant as far as the quantification accuracy and precision of MS1-based quantification. So if you download the latest version of our tools, the IonQuant and match between run and MS1 data is significantly improved compared to FragPipe 22 in previous versions. Now, again, as I mentioned at the beginning of my presentation, why I like single-cell proteomics communities, because people are trying all sorts of different things, right? Trying to find improvements, and we work a lot with Ingju, and he's a great collaborator, and he's trying a lot of stuff, as many of you here. And so, one paper that he approached us was on the TMT single-cell proteomics analysis that came out a few years ago now, where we all know how you do TMT with single-cell proteomics in that scenario, but what they were trying to do is to see if they can do everything in the linear ion trap and not using orbit trap. So to speed things up, to increase the throughput, sort of generate more data and get more IDs. And so we worked with him to support basically quantification on the linear ion trap data, basically low-res quantification in our tools. And in FragPipe 23, we also work, we basically added support for Astral TMT data, as well as team stuff, as well as TMT35 plex data. And I can tell you that one motivation for TMT35 plex, again, we have a collaboration with people using TMT35. And I'm super excited about Astral with TMT35, because that, I think, is going to be a super powerful strategy, including hopefully for single-cell proteomics data. So anyways, our tools now supporting Astral and supporting 35 plex data. Then others here, so Ryan was Kelly was working on, at the time, I guess, no longer interested in wide window acquisition, MS2, for single cell analysis. But we thought, okay, well, if people are doing that, how do we develop our MS-Fragger search engine that can most effectively, in the very, fast search MS2 data for wide window acquisition data, right? 4, 5, 10, maybe 20 Dalton window, but acquired as MS2 data. So not DIA, but DDA MS2. So Chimeris was capable of processing the data, but was extremely slow. So this work we published a few months ago on DDA Plus, where sort of single cell focus kind of faded in a sense because the field transitioned to other data acquisition strategies for single cell. But in applications where DDA MS2 is still useful, and there are plenty of applications, actually we found that people really love the ability of MS-Fragger DDA Plus to search MS2 data as a full window acquisition search, right? So identifying all chimeric peptides in that data acquisition window. That comes from our open search strategy that we enabled through the indexing in MS-Fragger. So we basically treat DDA data as if it was like a basically open search, in say 2 Dalton window. So anyways, we developed this full-fledged sort of DDA plus mode of MS-Fragger, and even though probably you don't see it applied now to single-cell proteomics data, as I said, there was a motivation, but now we have other uses for this strategy, and I think it's very successful. So here's a preprint of that work, and then on this slide here, we demonstrate that we are identifying a good number of identifications through this full isolation window search. And it's orders of magnitude faster than Chimeris, which was an alternative tool for processing this data. So another point I want to make, again, this is a work from Inju, who is at Genentech now, but they want to do a benchmarking study related to spatial proteomics analysis, where they looked at tissues and cutting the pixels out of those images, but they wanted to compare basically label frequentification and TMT strategies for TMT, MS2 based and MS3 based and various combinations. But how do you really compare different strategies if you use different tools? So actually FragPipe again was the only tool where we could do everything consistently, right? Everything would go through the same strategy, including percolator and full discovery rate scoring, whether it's a LFQ or even DIA. So I think the advantage of our pipeline is one advantage is that it allows you consistently compare different strategies that you may be applying as part of your optimization protocols or when you're deciding on what strategy to use. So as part of this work, basically, they benchmark different strategies and again, label-free with MS1-based quantification versus different TMT strategies. And FragPipe provided a comprehensive data processing and analysis, but most important for this, I think, application was that it was enabling consistent comparison between different data sets, right, between different strategies. So, if you're interested, just to summarize what they've done, is that, and that's an interesting paper because the conclusions are, of course, basically the main conclusion is that, the best method depends on what you're trying to achieve, but the label frequentification actually with MS1 based and match between runs actually offered a deep protein coverage, 3 to 4,000 proteins. Again, this is not a single cell, this is spatial proteomics analysis, but it had a really great dynamic range, and it was really great for quantifying specific markers. Of course, at the expense of a limited throughput, relatively limited throughput. On the other hand, TMT, MS2, and MS3 strategies were much higher throughput, but they had, a little bit more challenging as far as the quantification accuracy. And MS3 approach didn't significantly improve over MS2 due to lower sensitivity, but provided some other complementary information. So I think the conclusion is that, all of these techniques are complementary, and all of them have a role in the single cell proteomics study. But I think my point here is that by having consistent tools that we develop as part of this collaboration and previous collaborations I mentioned for single cell proteomics data, we have basically a toolbox that allows us to process data from any strategy in a consistent way. So the sort of this benchmarking becomes meaningful, and hopefully the results are good, and you would adopt them for your single-cell proteomics workflows. Now, as far as the DIA, data independent acquisition analysis, so we have a lot of things that we worked in the DIA area. So as you know, we were the first group that developed direct DIA strategy, and then sort of when everyone was thinking that DIA is a targeted proteomics workflow that requires spectral library building from DDA data and so on. So we had a tool called DIA umpire, and now we support all major workflows in FragPipe, and we still have our DIA umpire for certain applications. We still use it for immunopeptidomics data on orbit traps, for example. That's our HLA non-specific DIA workflow that uses DIA umpire. For orbit trap data, including astral narrow window DIA, we have a new tool called MSFragger DIA that does direct search of the data, followed by targeted sort of peptide-centric rescoring, all part of MSFragger DIA. But for dia-passive data, we also have now sort of the next generation DIA umpire called DIA tracer. So we have a tool for dia-passive data, and I'll show you one example in the context of, again, spatial proteomics data, how we can use this tool to analyze the data very effectively. But no matter what strategy you use, again, whether it's MSFragger DIA, DIA umpire, or just DDA data, what the advantage of our strategy is that everything goes through a consistent full discovery rate analysis. Everything will go through MS Booster for deep learning-based rescoring, followed by percolator and Q-value estimation, target decoy strategy. So everything becomes really comparable. It's not apples to oranges, right? Because everything goes through the same well-established workflows where, if you maybe choose some other tools to use, it's a black box, right? That may give you, 12,000 proteins in 5-minute runs, right? And of course, it's exciting, but is it real or not? It's a good subject for a discussion. So with all these workflows, we basically would build high-quality spectral libraries, and then we would pass those libraries for quantification, extraction of quantification, either to Skyline or Diane or Alpha DIA, another great tool that you will hear later in today's program. So, and one difference between, for those of you who are into DIA stuff, between how we approach things and the pure peptide-centric scoring is that the question is, when do you start using deep learning-based predictions? In peptide-centric strategy, you basically say, I can predict retention times at the beginning for the whole database, and I'm going to go to my data, and very, very complex data, with a very, very narrow retention time window, and only going to look on that retention time window and find something, and then kind of quickly generate a decoy and test there. And that's really a dangerous strategy, because you're not looking for modified peptides. You're not looking for other artifacts that are in the data. You just look for a particular unmodified tryptic peptide in a narrow retention time window that the software predicted for you. So that has a problem with the sort of FDR estimation. But also, it's a very time-consuming to do predictions for everything at the beginning if you do immunopeptidomics or PTMs and so on. So in our strategy, we do MSFragger sort of direct DIA search first, or DIA tracer followed MSFragger. And then we do predictions only for top-scoring peptides. And if the prediction is not good, so maybe the retention time is totally off because it's a modified peptides and so on, that's okay. It's just predictions not going to help us a lot in rescoring, but they're not going to penalize us too much, right? The bad prediction not going to penalize us and not going to create false positives, where if you kind of rely on the predictions at the beginning, that has an issue. And there are ways to deal with this, transfer learning and so on, but I'm just giving you sort of trying to give you a bigger picture here to understand all the complexities of different strategies for analyzing the data. So the sort of spectrum-centric first strategy that we are using in our tools has a lot of advantages when it comes to large sort spaces, PTMs, any other experiments where it's hard to predict peptides for the whole proteome accurately. So coming back to timsTOF data, I mean, this works really just beautifully how we did this, I think, because again, we take the dia-passive data and we convert it to a DDA-like data in XML files, right? We basically detect the precursors, detect fragments, correlate them, generate pseudo-mass spectrum, precursor fragment groups. Of course, it's all about signal processing, how you do it. It's really a lot of work went, actually years of work went into optimizing this just for ion mobility data, not to mention the early DIU work. But at the end of the day, when it works now is that you generate DDA-like data from your diapass of that data. And that data can be searched against using MassFrag or any other search tool. For PTMs, it can be searched in a regular way with PTMs and triptych, semi-triptych search, something again that's hard to do with other tools. And then you can, once you build the library, you can pass it to Diane, or the library when compatible with Spectronaut, or again Skyline or Alpha DIA. So you build a library, and then any tool can extract quantification from the original DIA files, from the single-shot DIA files using that library. And you can build a hybrid library as well. If you have DDA data or GPF DIA data, you can build a hybrid library. So in this particular example, we applied DIA tracer and FragPipe computational platform to allow input spatial proteomics data from the study listed here on this slide. And it's 148 microregions from 4 areas of human tonsil, right? And as part of the original work, they generated high input library containing 46 runs to generate high input library from a lot more starting material, and then use that library to do quantification from this single shot, single microregion data. So we demonstrated in our DIA tracer paper that came out a few months ago, is that we can basically, so yes, if we just look at the total number of proteins quantified, we would get less from a direct DIA method, only using low input data, compared to using the high input library. However, if you filter for missing values, basically saying that you quantify your data with a certain degree of completeness, for example, at 80 or 90% of completeness, the results are very, very similar already. And of course, as I said, the best strategy would be to build a hybrid library. So, if you do have high input data, and you have your single shot, low input, single cell, or like in this case, sort of the microregion data, why not build a hybrid library? Direct DIA coupled with in this case, the high input data was also DIA. So it's all direct DIA, but high input plus low input hybrid library. And then you do it all in one analysis with well-aligned retention times between all runs, very good high quality filtered library. So that, in our view, gives us the best results. So we were able to replicate the results of the original study here using DIA tracer and FragPipe computational platform on this timsTOF data set. And again, with just a few clicks, you can visualize the data using FragPipe Analyst as far as the differential expression analysis between different type of regions and the human tonsil. And Thixial cluster, this is a PCA plot, and a volcano plot, and known markers that differentiate between those different regions. All of this you could visualize and quickly validate just using direct DIA approach, only low input data, coupled with DIA tracer, MassFragger, a FragPipe, and then FragPipe Analyst at the end for visualization and differential expression analysis. Similarly, of course, we applied this also on Astral data. It works just fine. Actually, for Astral data, it works really well. It works well for all workflows, but for Astral, because it's a narrow window, it's really a great match to how we approach things. Except that, again, this is from Jasper Olsen lab, just some single-cell proteomics data from the Astral data. In this case, they used 20-cell boosting runs. And if you compare different tools, in this case, Spectronaut, Diane Library Free, or FragPipe with Diane, but only for quantification, you'll see that the numbers vary than the total number of IDs in the matrix file. But it's very misleading what each tool reports, because you have to look at how the data is filtered. And if you look at the number of proteins quantified without missing values, or with just a few missing values, a low percentage of missing values, you'll see that in our pipeline we actually get a really good results, because we control again the error rates in a run specific way very rigorously. And when you use these tools, just an advice for you to actually check how the data was filtered. For example, if you open the Spectronaut file, you will probably see that the Spectronaut results are filtered to 5% FDR in a run-specific way, right? Which is completely unacceptable for single-cell data, because you take the data built from 20 cells, right, in this case, transfer it to single-cell data, but apply only 5% FDR for the run-specific, right? So that's just wrong, right? So you always have to make sure that you consistently filter the data if you're comparing different tools. Just something that I always try to tell people. Not often people listen, but I keep saying that. And again, I mean, this is for a discussion, but the need of stringent filters, especially on ultras or astrals type of data, where if you actually, because we integrated everything with Skyline, that's beautiful because you could run FragPipe, just say run Skyline, it will generate Skyline document file, you can open it, and you'll see all these traces, and you'll get a heart attack if you look at this ultra or astral data, because you'll see that a lot of IDs have like one fragment here, one fragment there, they never coalute, and somehow it passes the Q values, right? So in our case, we prevent this from happening or try to minimize this type of magnifications by requiring that you identify a peptide by at least 4 or 5 fragments in the same MS2 scan, right? So we basically require that. And we also require by default that we detect MS1 signal. So we don't report, and you can then check it, but by default, you only will see IDs where you also found MS1, right? So this is we on purpose would rather report less IDs, but know that it's at least we detect the precursor, so we know it's not unmodified and modified peptides that somehow co-fragmented in the same window, and we're getting the shared fragments misidentified, and also making sure that at least there is some coalition of fragments, right? So I think that's important to think about when you do these comparisons, and if you start comparing the numbers, it gets into these listomics discussions, and I think we, as a community, especially the single cell community, whether it's still a race to show the highest numbers, as high number as possible, that we pay attention to how the data is filtered, and we make comparisons between different tools in a consistent way. And how do those comparisons, I don't know, the CVs are not really good metrics, and I'm not a big fan of multi-organism data sets, but I don't have a better suggestions to provide here, or significantly better. Now, so our strategy is to make sure that we provide visualization tools also to inspect the data. I already mentioned that we can build a Skyline document file, so that just you can generate them, and then you manually can inspect the data at least. And if you're into PTMs, and we work a lot with PTMs like glycoproteomics, we want to resolve between different glycoforms. So that's really great because you could actually visualize the data in different isomers and so on. Maybe less of interest for single cell proteomics data at this point, all this glycoproteomics and stuff. But or maybe it is, because I'm hoping that as the technology improves, single cell field will also go in the direction of PTM analysis and chemoproteomics and all this cool and exciting stuff that actually requires advanced searches of the data, looking for labile PTMs, unknown PTMs, and so on. Because this is, for my lab, is of great interest, right? We work on open searching and all these methods for a long time as a way to, MS-Fragger for me is like a dark matter telescope to really study all these unmodified peptides in this data. And with DiaTracer, for example, coming back to what I talked about a few minutes ago, you can generate MZML files, DDA-like files, and search them for PTMs in unconstrained way using MS-Fragger open search. That's one advantage of using DiaTracer MS-Fragger-based pipeline and FragPipe for DiaPassive data. So in summary, what I hope to tell you in this presentation is that FragPipe is a comprehensive proteomics platform that supports all sort of workflows, different quantification workflows, and many biological applications. And today, of course, I focus on single cell proteomics data, but we support a lot of different things. And our team is always open for cutting edge collaborations in all areas, including single cell proteomics, but not only anything that's challenging, that's really pushing us to further improve and develop the tools. And I think it's just, repeat what I just said a few minutes ago, that we need to focus on the quality of identifications, quality of the analysis, and not just the number of identifications coming out of these studies. So thank you. I will acknowledge my lab here and a few collaborators already mentioned, and Ingju in particular, since I used some of his work here today, and my lab. So thank you.
Great talk. I'm a huge fan of MS-Fragger open search. I know you didn't talk much about it, but I have a question on one of the different things that you mentioned. You said that you throw out identifications if you don't find a precursor. Do you have a sense of what proportion of the time that's happening?
Well, a really good question. Yes. So the question, yes. So we throw out identifications by default, right? You can uncheck that, but I would say that in Orbitrap's data, in Orbitrap DIA data, maybe you would get a at most a few 5% boost in number of IDs by accepting identifications where you didn't see any MS1 precursor. So not a whole lot. In Astral data, we actually get less. I think because the MS1 is such a, there's just so much stuff in there already, so much noise that having MS1 as a filter, okay, we don't see the precursor, we're not considered this peptide, right? Actually helps us, right? So we're not even losing, we're only gaining. And diapassive data, we are basically at the beginning, we start with detection of MS1 feature to assemble a precursor fragment group. So there we depend on detecting MS1 and that strategy. It doesn't have to be super rigorous MS1 detection, not like you have to see 5 isotopes with the right shape. We can relax the criteria, but we gotta see at least some MS1 signal.
Thank you.
So if I understand right, in FragPipe, the default DIA pipeline is to do something like DIA umpire, where you create a pseudo MS2 spectrum and then send that through. Did I understand that right?
For diapassive data. For diapassive data, yes. For Orbitraps, it's an option and not the default option. Our default option now is MS-Fragger DIA method, where we first search MS2 scan for all candidates without assembling precursor fragment groups, without what DIA umpire is doing. We flip the order of events. We first detect all candidates for each spectrum, like as if it was a regular database search. Then we take those candidates, create MS spectra, with the ions of the fragments, go and trace them in the data in a peptide-centric way. So that's our preferred strategy now. And DIA umpire only for certain applications like immunopeptidomics.
Alex, you are a very, very important person for this community here. I do believe that quality is really the challenge for the single cell now. But in the reality, the number of the fragment is getting less and less, even in the DIA. So I'm wondering, is it possible that you can use less number of the fragment ion, but combining with other factors, for example, the monoisotopic distribution, to increase the confidence of identification, instead of say 5 fragment is a minimum? Is that possible?
Yeah, I think everything is possible. It's we're going through a lot of these optimization experiments constantly, right? So we, for example, in MS-Fragger, we do de-isotoping first, the first step for high resolution data. We first de-isotope fragments and convert them to singly charged fragments, right? And we actually search the data only with the singly, we assume that we got with the de-isotope decharged the fragments, right? So we're not searching like 2+, 3+ fragments. We try to reduce the search space. So in a sense, like we're matching less fragments, but the higher quality fragments in a way, right? And it tends to help us. So for single cell data, again, I think more optimization needs to be done, and things are really improving really fast, and how you generate the data and the quality of data is changing. So I think it's still kind of a moving target, but yes, we are going through steps like this always to see how we can improve the tools.
Sorry, one more question. Today, like Ryan talked about the very fast gradient, like 0.5, different kind of short gradient. So in the DIA computation, for this kind of different gradient, do you think the spectral library can be universal? By using the reference peptide like LRT for calibration, is that robust enough?
Well, I think it's a really complicated question. I'm not able to give an answer in 5 seconds. I'll just comment that if you build a sample specific libraries from your data, you're always going to be the best libraries than relying on something that you build from very different gradients or from bulk data, right? So that's why we prefer building direct DIA plus some data that's generated on the same instrument, they say in the DDA mode. But I think with short gradients, the more interesting question to me, or equally interesting, is, should we do quantification from MS1 data, for example, right? Identify from MS2 or quantify from MS1. In our pipeline, we try to report both types of quantification. And I think with something like Astros, or Ultra, probably the same, short gradient data, the quality from SMS spectrum is not going to be super great. You can still identify the peptide because you can build a good library, but MS spectra are not great. But because the signal split between many, many fragments, but MS1 often looks better. So we prefer to actually quantify from MS1. And again, in our pipeline, we rely on observation of that MS1 signal. So yeah, there's a lot of things to compare and contrast. It's yeah.
Alexey, I think your point about focusing on quantitative accuracy rather than identifications is very, very resonant with many of us. Certainly, I strongly agree with that. And you discussed the challenges with defining metrics for it. It is very difficult, but I think unless we have metrics, it will be very difficult to shift the focus. Whatever is measurable, people optimize. If something's not measurable, it is difficult to advocate for it. So I think it is very important for us to, as a community, to discuss what are these metrics that we are happy with so that we can replace some of the low bar metrics such as consistency of detection or repeatability as quantified by CVs. I agree with you that that's not a very good standard for anything. We can be consistently misidentifying the same signal, consistently misquantifying it, and getting exceedingly low CVs. It's just not a good metric. But we cannot only say what doesn't work. I think we need to find the time to have the discussion of all of the metrics that are deficient in some ways, which are the metrics that are the most useful, and advocate for those, recognizing that they may not be ideal, but still we have to argue for them. I'll make one suggestion. I'm not saying that this is the ultimate and the best. I think this really has to be a wider discussion, but one metric that resonates with me is the consistency of quantification of the same precursor from different scans in, for example, Plex DI data. We normally have multiple MS1 level scans, multiple MS2 level scans, so it can allow us to quantify relative abundance changes across multiple Plex DI samples. And I think the consistency between those can be a useful metric. But I'm also very interested to brainstorm with you and with other people in the community and to see what are these metrics that, as imperfect as they are, we want to advocate. Because if we don't advocate for those, people are just going to use CVs, whatever is the easiest thing to use, the lowest common denominator.
I would also add to that I think the importance of how you post-process the data. So we had a tool, MapDIA, actually, that everyone forgot about, but that was a tool in the early days of DIA that would basically correlate fragments to each other across multiple runs and to the precursor to the peptide and even to the protein level as a way to throw away the outlier fragments and peptides that basically show discordant pattern. And as a way to find high quality fragments and use only those for quantification. Of course, there's a challenge that is actually maybe biologically relevant, isoforms that, and things like this, but as a filter of low quality data, that was really great. So I guess what I'm trying to say here is that, once you quantify your fragments and peptides, what you do next is equally important. And that's sort of a little bit underdeveloped area at the moment. There was an effort before and kind of now, I think it will come back to sort of looking at post quantification, sort of like the next steps for filtering out, finding the right fragments to use for peptide and protein level quantification up from the initial search.
So let me ask another. I have many questions, so just ask one more so that there is time for others. And this one is more of a challenging question, but curious what you think about it. When we do MS1 level quantitation, certainly in DI data, one of the main sources of error is interference, and that interference tends to come from highly abundant ions nearby that we also quantify. And we usually throw away the contaminated signal, but there is another possibility, which is maybe we can try to estimate that interference because we measure both the source from which it's likely to have come or potential sources, and we measure the ions that have been interfered with. Do you think it is practically feasible to think of ways to model that and at least partially diminish the effect of that interference?
So it's a good suggestion, an interesting idea to pursue. It's a challenging one. I'll give you an example, a simple example where it's actually turned out to be very challenging, like TMT data correction for the isotopic impurities, right? Sounds like a simple problem. You have so much of this channel leaking into this channel, right? And you solve the linear equations and you correct for the data. Well, it turns out that if you do that, despite what maybe Thermo would tell you or others, that actually you make things worse, because for low abundant peptides, where you have the really challenging low abundant data, you start correcting for these things and you make things worse. It works beautifully for high abundant peptides. You can demultiplex, correct for all of that. And then you start getting outliers and you look at them and it's a low abundant signal, you overcorrected, it created differential expression. So it may work really well for good, maybe even for 90% of the signal. The question is, will you start creating artifacts in the data for something at the noise level that perhaps what you're most interested in, accurate quantification of low abundant things. And this is where, unless you do it, you wouldn't know how that would work, right? So, but yes, it's an interesting direction to think about.
Thank you.
How to deal with this shared peptides, like how this tool will consider these shared peptides and how to give weightage for this.
Oh, this is my favorite paper, and it's 20 years now that I wrote a paper called the protein inference problem, right? That talks exactly about that. So we use a tool called protein profit in our pipeline. Again, no matter what, whether it's a DIA, DDA, SciLac, whatever, everything goes through the protein profit, and we assign peptides to proteins with most evidence through the protein profit algorithm. And that's how we approach that. And we have this razor assignment, and most other tools do that. And we use DDA strategies for DIA data as well. Now, what's done in other tools, they say that they also do protein inference and try to sort of assign peptides to protein accession numbers and genes in a consistent way. But it's a little less known what exactly those tools are doing. But yes, no matter what, we have to deal with this protein inference problem of shared peptides. And we often in proteogenomic studies, and we do a ton of proteogenomics as part of CPTAC consortium, we often end up summarizing to the gene level, not to deal with the problem, especially if you integrate with RNA data. Of course, if you really need to resolve between protein isoforms and so on, you have to deal with this problem, and it becomes a bigger issue, and you have to find what works for you. But at least in FragPipe, we have a consistent way to assign peptides to proteins. Consistent given the evidence that we have. Unfortunately, we often don't have enough evidence, and we just have to present it to the user in the most transparent way possible, the ambiguity.
I wonder about prediction. So it's relatively easy to predict retention time, cross-section, or fragmentation of MS2. And I wonder how useful it would be to be able to predict MS1 intensity, so we know which peptide from a given protein should be observable, and we don't see them, we know they are modified. And can we also use this information for scoring and other things? I wonder what do you think about MS1 prediction?
Yeah, great. Parag Malik, he was my friend. He was a postdoc at the same time as ISB as I was in Rudy Abrasov lab. So Parag had a paper 20 years ago also on the flyability and trying to predict what peptides would be observed. And others also worked on that. So, I mean, there are several tools, and we tried actually incorporating in MS Booster a prediction of whether a peptide will be observed or not. So it's not exactly the same as predicting the intensity, but predicting whether you'll see the peptide and sequence it, right? So it correlates, of course, with the MS1 intensity of the peptide. It had a minimal impact using the tools that we tried at the time, which is a few years ago. I see new tools coming out, and maybe they will benefit the analysis, but I think it has to be a certain type of transfer learning applied, because you can't trade it on gazillion of data from average proteomic study, right? You kind of see what proteins expressed in your particular study, and maybe based on that, figure out what should be the abundances of proteins that you have questions about that's not really clear identifications, right? So I think there is something to that, but it has to be in the context of transfer learning that looks deeper in what you can confidently identify in your study. Learn from that, predict the intensities or observability of proteins in that particular sample, right? For the questionable ones that would benefit from this rescoring from the predictions. So something along those lines may work.
Well, let's thank our speaker again.
Okay.